This experimental system aims to characterize the contribution of specific proteins to the invasive process. Begin with retroviral transduction of cells with the fluorescent protein of interest. Also, create the 3D matrix of matrigel protein in transwell inserts. And prepare a cell suspension at the appropriate dilution. Using the matrigel transwell system, measure the transmigration response to the chemoattractant with test drugs or treatments. Proceed to visualize and analyze cell invasion by confocal microscopy. Ultimately, results can determine the magnitude and mode of cell invasion into three-dimensional protein matrix.
The power of this technique over existing methods like the traditional transwell invasion assay is its capability to derive both quantitative and qualitative data on cell invasion into a three-dimensional protein matrix. My collaborator, Diane Crighton, will now demonstrate the method.
Culture retroviral packaging cells in DMEM containing 10% FBS for 48 hours. Transfect the cells with retroviral DNA according to the manufacturer's instructions. Incubate for 24 hours. Rinse the wells twice with medium, then add 1.5 ml of 10% FBS DMEM per well and incubate for 48 hours. To collect the packaged retrovirus, transfer the tissue culture medium to 2 ml microcentrifuge tubes. Centrifuge to pellet out any cells and harvest the supernatant into a clean tube. Store this retrovirus stock at -80 degrees Celsius. Now to retrovirally transduced cells, remove media from an overnight culture and add 1 ml of retrovirus stock supplemented with